Move on to the last talk of this session by Shuai He. So Shuai, can you share your screen? So Shuai He is a PhD student in Sun Yat-sen University Cancer Center from Professor Jinxin Bai's lab. So let's hear their work on COVID-19. Yeah. Thanks. Yes, you can start. Can you unmute? Can you turn on your microphone? It's muted. It's muted. You unmute. Yeah, now it's all. No.
Okay.
Yeah,
okay.
Go ahead.
Thanks for Chair Zhang's introduction. Good afternoon, everyone. My name is Shuai He, a PhD student in Sun Yat-sen University Cancer Center. It's really my honor to be a selected speaker here to share my work with you. My topic is single-cell transcriptome profiling and adult human cell outlines of 15 major organs. This work is under the direction of Professor Jinxin Bai and Zhiyong Guo. As we all know that human body is very complex and heterogeneous system, which consists of different organs and cells. Although recently, scientists have already achieved remarkable findings in exploring the heterogeneity of human cells. However, a systematic and comprehensive single-cell transcriptional profile across multiple organs of a normal human adult has been pending. Thus, our aims were to fully decipher the heterogeneity of human cells, including cell composition, gene signatures, null cell types, and regulatory networks in different organs. This is the outline of this topic. First, we collect 15 organs from one adult and dissociated the samples into single cells. We used the FACS to select the variable cells in each tissue and then performed single-cell mRNA, TCR, and BCR sequencing. In totally, we obtained more than 80,000 cells. Next, we manually annotated the cells in each organ and obtained 252 cell subpopulations. As the large number of cells we obtained, we could identify null cell types, including CUCH positive fibroblasts, specifically distributed in the skin, and fibrous smooth cells, marked by co-expression of MMP2 and ACTE2, which broadly distributed in multiple organs. We further confirmed our findings in existing datasets, such as we observed similar signature genes in both our and published datasets for CUCH positive fibroblasts. We then confirmed our findings in multiple donors using multiplex multiplex immunofluorescence. We then merged all cells from 15 organs for global clustering and identified 43 major cell subtypes. As expected, cells from same lineages were grouped together, including B cell, NK cell, T cell, fibroblasts, endothelial cells, and so on. While epithelial cells exhibited large diversity and were clustered according to their origin, such as shown in the T-SNE, we could see we could see epithelial cells was isolated from others, such as in esophagus, small intestine, and skin. Immune cells play important roles in many aspects and consist a large part of all the collected cells. Thus, we first investigate the heterogeneity of immune cells. Here, give T cell and B cell for examples. We observed most T cell clusters showed a distribution at an organ-specific manner, especially for T-random memory T cells. We observed that T-random memory T cells were clustered according to their origins, shown in the T-SNE. We then try to explore the potential mechanism of the diversity within T cells. We evaluated the activity of TFs. Interestingly, we observed cluster-specific activity of TFs for both CD4 and CD8 T cells. In addition, we found AP1 dimers had specifically highly activity in both CD4 and CD8 TIM cells. We then confirmed the co-expression of CD8A and C4s in T cells. We could say C4s and CD8A co-expressed in multiple organs. As all the samples from one donor, which offer us an opportunity to explore the clonal structure of TCR. Interestingly, we observed clonal expansions was much commonly for CD8 T cells in different organs. The higher diversity of CD4 T cells in different organs and the broad links of TCR within CD8 T cells across organs. For B and plasma cells, interestingly, we observed similar activities of TFs within B and plasma cells, but distinct between the two in both our and Hans dataset. For non-immune cells, here we give epithelial cell, for example. We observed that epithelial cells exhibited large heterogeneity, inter and intra-organ in both our and HCL dataset. That was Hans dataset. Although the large diversity of epithelial cells, we observed the cells from digestion and non-digestion derived organs were close clustered in both our and HCL datasets. Then we found the cells from digestion and non-digestion derived organs had similar TFs activities, but distinct between the two, as shown in the box in the two datasets. This observation might partly explain the similarity of the gene profiles within the two groups. Since we observed heterogeneity of cells in each organ, we explored their potential intercellular communication networks, inter and intra-organs. We observed broad interactions inter and intra-organs. Well, we observed CD8 T cell, myeloid cell, epithelial cells had the most intensive interactions with other cell types in both inter and intra-organs, indicating that these three cell types were the core of the cell-cell interactions. We then summarized our findings and provide an interface-friendly web tools for users.
So it's already 10 minutes.
Yeah. I will quick go. Okay. In summary, our study decode the heterogeneity of cells in the human body, provide a deeper understanding of the biology mechanisms related to health and disease. our data set has many usage, such as we could quickly overview the ACT2 expression across different tissues. At last, I thanked my colleagues who make the equal contribution to this work. Yin He Wang for collection the samples and figuratively tailoring, Yang Liu for collection for data analysis and sample preparation. I also highly appreciated my supervisor, Jin Xinbei, who offered good help during this work. This work is under the revision of genome biology. Thank you for your attention.
Thanks for the talk. So we have 2 minutes for questions and answers. So I have a question about you mentioned that inter-organ cell-cell communication. What do you mean by that? So if the cell types are of different organ, how do they interact with each other?
We choose cell. We choose other cell. We evaluated the ligation and receptor expression between any two cell types and across in different organ. And these two cell types, the one must be immune cells.
Then what do you suspect is the mechanism that the cells interact across organs? It's kind of through some fluid, or what is the underlying mechanism?
Because some cell types, such as epithelial cells, they couldn't move across organ, but other cell types, such as immune cells, they could move across different organ, and we evaluate the interaction between immune cell, mainly immune cell and non-immune cells.
Okay. Yeah, go ahead.
I think the mechanism maybe the cells could produce some chemokines or some other protein.
Okay. So I think time is up. Thank you, and can you stop sharing your screen?
Okay.
So thank Shuai for the talk, and thank all for speakers. So, and thank for the audience for participating in this session. So now we conclude this session, and we will have a short break, about 12-minute break before the keynote speaker by the keynote speech by Shuai He. So let's come back in 10 minutes. Not this room, in the plenary room, in the plenary meeting room. Okay. So see you there.